Okay, moving on, finishing up chapter 7. This is actually kind of complicated, so bear with me here. Okay. Okay. So acetylcholine. Okay. So the most common neurotransmitter, and the neurotransmitter that everyone first learns about is acetylcholine. And you probably learned that in anatomy already. It binds muscle and causes muscle contractions and skeletal muscle. Okay. Acetylcholine is a neurotransmitter that directly opens ion channels and binds to the receptor. Okay. In some cases, ACH is excitatory. In other cases, inhibitory. Okay, depending on organ involved. Okay. Okay. It's excitatory in some areas of the CNS, in all the somatic motor neurons, so the skeletal muscle, and in some autonomic motor neurons. Okay, it's inhibitory in some autonomic motor neurons. Okay. Okay. There are two types of acetylcholine receptors. Okay. Nicotinic receptors are the ones that can be stimulated by nicotine. Okay. And muscarinic ACH receptors, which can be stimulated by muscarine. Okay. Okay. These, the nicotinic receptors are found in the motor implant skeletal muscles, in the autonomic ganglia, and some parts of the CNS. Okay. The muscarinic are found in CNS and plasma membrane of smooth and cardiac muscles and glands. Okay. Okay. So some terminology that you guys have probably heard but don't know the exact meanings of. Agonist and antagonist. Agonists stimulate the receptor. Okay, so they're like a fake neurotransmitter. Okay, nicotine can stimulate nicotinic ACH receptors. That's why people smoke and like nicotine. Muscarine from muscarinic ACH receptors. That's why it's a poison. Okay. Antagonists are drugs that inhibit a receptor from working. So they bind to the receptor, but they don't activate the receptor. They prevent it from working. Okay. Atropine is an antagonist for muscarinic receptors. Curare is an antagonist for nicotinic receptors. Okay. Hold on 1 second. Oops. Okay. Antagonists are drugs that inhibit a receptor. So the way I like to think about this, the drug RU46 is an antagonist for progesterone. That's what causes spontaneous abortion. Okay. Okay, the binding of a neurotransmitter to receptor can open a channel in 1 or 2 different ways: a ligand-gated channel or a G protein coupled channel. So the nicotinic ACH receptor is a ligand-gated channel. Okay, so you can see it here. Okay, when ACH, there's 2 sites. Okay, they bind, and this opens up this channel. This channel simultaneously allows sodium to come in and potassium to rush out. Okay. This is an electrochemical gradient. More Na plus flows in than K plus out. Okay. 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 This inward flow of sodium depolarizes the cell, which causes an EPSP. So it excites the cell, right? increases the risk, the chance of firing. Okay. Okay. EPSPs from binding several acetylcholine molecules can be summed up together. Okay. So the more ACH can activate more channels, bigger EPSP. Okay. Remember, EPSP is not an all or none response; it's a graded response. Okay. So the more EPSPs you receive, the more likely that neuron is going to fire. Okay, leading to the action potential. Okay, so this is one way of actually getting enough like I said, one EPSP is not enough to get it over a threshold. You have to have multiple EPSPs to actually get there, and therefore you can sum up EPSPs. Okay. Okay, this is a comparison between EPSPs and action potentials. I would definitely spend time going over this. Okay. Okay. The other way acetylcholine works through the muscarinic receptor. Okay, it works through G proteins. G proteins work on downstream things. So now it depends on what downstream things you have to make it either a plus or minus. Okay, that's why the muscarinic is either excitatory or inhibitory, while the acetylcholine, the nicotinic is always going to be excitatory. Okay. Okay, this is, let me show you, let me explain the picture. This is easier to explain. So acetylcholine binds to the receptor. The receptor then activates the G protein. The G protein subunits actually bind. So they dissociate here. The beta and gamma subunits actually bind to potassium channel right here, and open potassium channel, and this is an inhibitory response. You have excitatory responses when it opens up sodium channels. Okay. Okay. Or closing K plus channels. Okay. 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 Acetylcholine esterase. Okay. So acetylcholine is released, okay, as a neurotransmitter. And all neurotransmitters, you want to destroy the neurotransmitter as soon as you release it, because you want one action potential. You do not want continuous action potentials. Okay, so what you do is you have an enzyme that breaks down acetylcholine right away. Okay, so, and that's acetylcholine esterase. Okay, so as soon as it's released, you want to break it up right away and have it taken back up. Okay. Choline esterase inhibitors are drugs that block the acetylcholine esterase, which increases the amount of ACH in the synaptic cleft, enhances the synaptic transmission. Okay. All right, let me find here. Let me just go through this. Okay. If I leave the acetylcholine there without destroying it, I get constant stimulation. Okay. And that actually is what nerve gas is, and stuff, and other things. Okay. Assuming there are cobra venoms that do this, and stuff where they they bind to the ACH and then, or they prevent the enzyme from breaking down ACH. Okay. All right. Okay. Monoamines are regulatory molecules that are derived from amino acids. So catecholamines are the include dopamine, norepinephrine, epinephrine. I'm sure these are things that you guys have already heard of. Serotonin is derived from tryptophan. Histamines are derived from histidine. Okay. Like ACH, monoamines are made in the presynaptic axon. They're released via exocytosis through the same way. They diffuse across the synapse, bind to the receptor, and then they're also broken down and reuptake by the presynaptic cell. Okay. Okay. So exact same thing, right? As acetylcholine that we learned earlier. Okay. Okay. So what can you do? Well, you can do the same thing with acetylcholine. You can block the reuptake. Okay. Or block the degradation. Okay. MAO inhibitors have proved useful in the treatment of depression, panic disorder, Parkinson's disease. Okay. Okay. So the monoamine action, all of them use a G protein and a cyclic AMP second messenger. So here it is. So what happens is this is just norepinephrine, but all the mechanisms are the same. They bind to the receptor activates the G protein, which dissociates the alpha subunit binds to adenylyl cyclase, which activates ATP into cyclic AMP. Cyclic AMP is a second messenger, and that will activate a whole bunch of other things downstream. Okay. Okay, serotonin as a neurotransmitter is implicated in mood, behavior, appetite, cerebral circulation. The drug LSD are agonists of serotonin. Also, serotonin specific reuptake inhibitors are used to treat depression. Prozac, Paxil, Zoloft. I would like to add the personal ecstasy is a SSRI. It prevents serotonin reuptake, so you have increased amounts of serotonin, which increases your mood. Okay. The downside is after that ecstasy wears away, there's decreased serotonin levels in your brain, and that leads to usually depression afterwards. Okay. Okay. So not advocating taking E, by the way, or ecstasy. Okay. So, okay. Dopamine. Dopamine is so in two main regions. This is involved in motor control, and I'm sure you guys already watched. There's a very famous movie out there where dopamine trials and stuff. They give them L-Dopa. So dopamine, for some reason, doesn't get across the blood-brain barrier. L-Dopa does, but L-Dopa, your body converts, your brain cells can convert to dopamine, so you get increased amounts of dopamine. Okay. So Parkinson's disease is caused by degeneration of these neurons, so you treat it with L-Dopa. Okay. 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 Schizophrenia is associated with too much dopamine, so you block them out dopamine. Okay. Okay. Okay, norepinephrine stimulates PNS and CNS. So they're used by neurons in the brain that associate with arousal. Amphetamines work by stimulating norepinephrine pathways in the brain. In this case, arousal is not sexual arousal, is that it's alertness. Okay. Okay. I'm gonna skip the part about neurotransmitters. They're all just really straightforward. So moving on to okay. Okay. Synaptic integration in the neural pathways. So okay. Here. Okay. So these neural pathways, they add up. So what is happening here is you have two ways of getting to threshold. Okay. One is multiple signaling coming from multiple axons, all with the EPSP. The other way is multiple times that same axon sends the EPSP. Okay, so that is either spatial summation, multiple axons, or temporal summation, one axon multiple times. Okay. Okay. Repeated use of a neuronal pathway may strengthen or reduce the synaptic transmission. Okay. When repeated stimulation increases excitability, it's termed long-term potentiation or LTP. Okay. 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 And then that's memory. Okay. Okay. I'm gonna skip this part. So again, what you have competition between the IPSP and EPSP, and therefore you can't reach the action threshold. Okay. Okay. The calcium channels are so prevention of presynaptic inhibition. Okay. Sometimes the neuron synapses on the axon of the second neuron, so and it can cause prevention the IPSP, which prevents the release of the excitatory neurotransmitter. So by deactivate, remember the signal has to change into a calcium signal at the very end. So if I block, then and that calcium channel is voltage gated. So if I block the voltage gated calcium channel from opening, then I block the release. Okay. Okay. All right. Hopefully that makes sense to you guys.